Hello, I am Dr.Ashok Kumar, Head Department of Zoology, Ganga Singh College, Chapra, running under JP University, Chapra, Bihar. In my last class, I had explained the protozoan diseases caused by protozoan organisms in fishes, in which two types of diseases. First was ichthyopteris caused by ichthyopteriasis. Disease name is ichthyopteriasis caused by Ichthyopteris multiplicus. So it's Ichthyopteris multiplicus, and in which I had already stated in the life cycle that there are four stages. That is, the therions infect the dermis as well as infected fish in the skin. Nodules like structures they appear, and these nodules, these trophozoites come outside and attaches with the substratum, and these are called adult or tomotes, and these tomotes divide to form these tomotes divide inside the cyst. The tomotes are encysted. It remains inside the envelope, and the tomotes convert itself into a large number of tomites, and these tomites breaks the cell or cyst of the tomotes, external cyst of the tomotes, and now it becomes free as therions. Now they remain in search of a new host. If new fish comes, or uninfected fish comes, under that condition, it penetrates, it attaches itself with the skin, and then goes inside, and again it repeats its life cycle. So the therions are infective stage. Now the treatment of the control is only possible on the life stage outside the dermal tissue of the host. The stage of Ichthyopteris multiplicus within the dermal tissue of is quite impossible, almost impossible to remove using chemotherapy. Hence, without injuring the host, and the measures can be taken. That is, after netting, we separate the diseased fishes and give hourly bath in 1:5,000 solution of formalin or formaldehyde solution for 7 days. First thing is this, and second, sodium chloride bath in 2% sodium chloride solution for 7 days or more. Then the fish becomes out of stress as the parasite dies. Hence, this is all about the treatment of the ichthyopteriasis disease. Now, trichodiniasis is also is caused by Trichodina and Tripartiella disease, Tripartiella genus of protozoan. Trichodiniasis is caused by Trichodina as well as Tripartiella genus. There are two species, that is Trichodina reticulata and Trichodina nigra, and 3 species of Tripartiella, that is Tripartiella copiosa, Tripartiella obtusa, and three diseases. They infect the carp skin and having miniature spiked gills with cilia. Due to the sucking action of these parasites, the epidermal cells of the host are irritated. Due to sucking action of these, they are disc-like, and due to the sucking action of the skin or gills, the epidermal cells are irritated, and under irritated condition, they secrete a lot of slime. A lot of slimes are secreted after being irritated as they suck and gets cytoplasmic constituents or as their feed as their food, they get it from the and the skin as well as gills, they become irritated, and as a result a large amount of slime are secreted out of the epidermal cells of the host fish, and ultimately the fish die. The fishes die. Hence the parasite then feed on after the death the parasite Trichodina and Tripartiella. Both they now they feed on the dead cells of the fish. But when fish die, then feed upon the dead cells, and asexual reproduction occurs mainly by binary fission, and hence Trichodina and Tripartiella, both genera, they become Trichodina and Tripartiella, they grow after eating the tissues of the host fish. The species now Trichodiniasis. Which species? The fly and fingerlings of Katla, IMC, as well as exotic carps, as well as other forms. So IMC, Indian major carp, Katla, the species affected by Trichodina, these are Trichodina, the species affected by Trichodina and Tripartiella are IMC, means Katla, Lebiorohita, Sirhaina mrigla, and exotic carps like silver carp, grass carp. Silver carp means Hypophthalmictis molitrix. Grass carp means Ctenopharyngodon idella, and common carps means Cyprinus carpio. Besides these carps, Tilapia mozambica and other cultured carps, they are these species are affected by the action of Trichodina as well as by the action of Trichodina and Tripartiella. Trichodina and Tripartiella species, that is Trichodina species are Trichodina reticulata, Trichodina nigra, and 3 species Trichodina bulbosa, Tripartiella bulbosa, Tripartiella copiosa, Tripartiella obtusa. These 3 species of Tripartiella as well as 2 species of Trichodina, they they remains as parasite on the body as well as gill of the fish, of the carps as well as tilapia and other cultured carps. Effect on the host. Effect on the host is hyperplasia and hypertrophy of the gill filament occur with proliferation of mucus cells. Hyperplasia means increase in the size of the organ or of the tissues, increase, and hypertrophy means the increase in size by eating the nutrients of the gills as well as skin, and the proliferation as a result, mucus cells proliferates and their number increases. Mucus cells lying on the gills, they proliferate, and it means their size increases and proliferation of mucus cells. And the respiratory function of the gill lamella is hampered because of the proliferation of the gill epithelium. So the respiratory function is hampered, often resulting in asphyxiation. As asphyxiation means no oxygen is taken, or that condition is asphyxiation, in which suffocation occurs due to the lack of the oxygen. Hence, it causes, as it attacks on the gill epithelium. Hence, asphyxiation is seen there, and the fish becomes very irritated. So, effect on host is hyperplasia and hypertrophy of the gill filaments, and occur with the proliferation of mucus cells. Number of mucus cells become more. The respiratory function of the gill lamella is hampered, and hence it results in asphyxiation as per Das and Pal in the year 1985, and hence Das and Pal in the year 1989. Causative agent, as I had already mentioned, that is Trichodina, two species of Trichodina, that is Trichodina reticulata and Trichodina nigra, and three species of Tripartiella, Tripartiella obtusa, Tripartiella bulbosa, and Tripartiella copiosa. These are the three species of Tripartiella and two species of Trichodina, that is Trichodina reticulata and Trichodina nigra. Now, causative agent, that is Trichodina reticulata, Trichodina nigra, Tripartiella bulbosa, Tripartiella obtusa, Tripartiella copiosa. These are the five ciliates, five species of ciliates. Morphology. Now, the structure of the Trichodina, that is, the shape of the body varies from flat disc to bell-shaped. Flat disc to bell-shaped. So it varies. It is concave on its aboral surface. And it attaches to the host surface by means of adhesive discs. It attaches the host surface by means of adhesive disc constituted by skeletal elements. The disc consists of a ring of the disc consists of the ring of denticles, which has a central part as well as central part and centrifugal and centripetal projections called blade and thorns. respectively. So centrifugal and centripetal projections are found there, and they are called blades, centrifugal blades, and thorns. Centrifugal and centripetal projections are called blade and thorns respectively. Means centrifugal projections are called blades, and centripetal are called thorns. The denticles are held to one another. The denticles are held to one another. Like in this, if there is depression, the second denticles will remain here fit, and the third will remain here as in this way. So the denticles are held to one another by inserted conical parts, around which is a ring of fine skeletal rods called radial pins. The denticles are held together. Suppose one denticle is in this way, so and here, and the other remain in this way. So these are called radial pins. The disc is encircled by a movable border membrane. The disc remains encircled by a movable border membrane, and this border on the periphery, it remains encircled by border membrane, and these are reinforced by fine skeletal ridge. The border membrane seals of the attachment. The border membrane seals off the attachment disc help to maintain the suction. The fine skeletal ridge. The border membrane, which remains provided with the border membrane, that remains provided with the skeletal ridge, and that maintain the suction. That this due to this suction, this is skeletal. So skeletal ridge, they maintain the suction that temporarily attaches the ciliates to the host surface. The ring of denticles, that ring of the denticles, and one denticle, suppose this way, one denticle here broader, and then this is conical. So here again now the next in this way. So the ring of denticles and radial pins located above the arboreal. The ring of denticles and radial pins located above the arboreal surface, or arboreal surface and pellicular surface provide rigidity to the cells. These allow elevation of the center. Of the attachment discs. So they become, so in Bell's condition, these allow elevation of the center of the attachment disc via a complex system of myofibrils. Apically above the border membrane, there is a ciliary sheath of single row of cilia. There is a single row of cilia. These are covered typically and these are covered apically by the velum. Velum is a membrane. So the non-contractile oral surface lacks a. The non-contractile oral surface is non-contractile, and that lacks a separate epistomial disc. The non-contractile oral surface is not contractile, but arboreal surface is contractile, and hence the and hence it lacks a separate epidermal epistomial disc. So three things are here: denticles, and then around the denticles there are radial. These are denticles one by one, and then radial pins, and after radial pins there is a there are a skeletal ridge, and border membrane contains a skeletal ridge. With life cycle the arceolariid ciliates, that is Trichodiniasis tripartita, they reproduce by binary fission, and the adhesive disc separates in two. The adhesive disc separates in two semicircles, which then close to form a small. They become the daughter individuals. So how it is treated? Treatment, water quality treatment. In next class, I will explain about the treatment of the trichodiniasis. Thank you.